Hi guys, so today I'll be going over mitosis and meiosis and briefly describing the steps that occur during each stage. So let's talk about mitosis first. Now the interphase stage is a growing stage for the cell before it enters cell division, and it's also when the DNA duplicates. During the interphase stage, it's also divided into three substages: G1, the S phase, and G2 phase. So G1 and G2 phases are both growing stages. The cell grows organelles for its cytoplasm, and it also synthesizes proteins. And the S phase is when the DNA is duplicated. And then interphase always precedes prophase. So after interphase, you have the cell division, which can be either mitosis or meiosis. So during the prophase stage in mitosis, we have centrosomes, which consist of microtubules. Centrosomes separate from each other because the mitotic spindle begins to form during prophase. The nucleoli, they disappear, and the nuclear envelope also starts to break down. By late prophase, you actually have microtubules interacting with the chromosomes, so they are attached to the chromosomes at the kinetochore. And also, one important thing during prophase happens is that the chromatin, it condenses into visible chromosomes. So they're very thick, and as I talked about in my last video, the DNA molecule wraps around the proteins and it forms visible chromosomes, so they're easy to observe. Now, next stage is metaphase. During metaphase, the chromosomes align along the metaphase plate and they are attached to opposite poles of the spindle. The next stage is anaphase. Anaphase, sister chromatids, they separate and move towards opposite poles. So sister chromatids are actually attached at the centromere, but during anaphase, they separate and move towards opposite poles of the spindle. And the last stage is telophase, followed by cytokinesis. In telophase, you have two daughter nuclei form, the nuclear envelope reforms, nucleoli reappear, the chromosomes decondense, so they get longer and skinnier, and you have two daughter cells that are genetically identical to each other and the parent cell. So both diploid and haploid cells can undergo mitosis, but only diploid cells can undergo meiosis. And this is what occurs in mitosis. Now let's go through meiosis. So meiosis is also, the first stage is basically interphase, which functions the same in meiosis. It's followed by two stages of cell division, meiosis 1 and meiosis 2. So in meiosis 1, you have prophase 1. Prophase 1, your chromosomes decondense, centrosomes are separating, your meiotic spindle is beginning to form. And also additional things happen during prophase 1 of meiosis 1. So during this stage, the homologous chromosomes, they pair up with each other. So homologous chromosomes, remember, they are a pair of identical chromosomes. They are the same size, they have the same centromere location, and they carry genes controlling the same inherited characteristic. So the homologous chromosomes, they pair up, which is called synapsis. The genetic information is exchanged between the homologous chromosomes, and what forms is called chiasmata, which are formed during the crossover. So this is all happening in prophase 1 of meiosis 1. Now in metaphase 1, the homologous chromosomes align along the metaphase plate and they are also attached to the opposite poles of the spindle. During anaphase 1, the homologous chromosomes will separate and move towards the opposite poles of the spindle, but the sister chromatids still remain attached at the centromere. The sister chromatids will separate during meiosis 2 in anaphase 2, but in anaphase 1 of meiosis 1, only the homologous chromosomes are separating. And then we have telophase 1, in which each half of the cell has one complete set of chromosomes and two haploid cells are formed. So remember, only diploid cells can undergo meiosis, and they produce two haploid cells after meiosis 1 has been complete. Now we will go through meiosis 2. So in prophase 2 of meiosis 2, your meiotic spindle, it reforms again, and you still have a pair of chromosomes, a pair of sister chromatids. They're still attached at the centromere. Meiosis 2 is basically the same thing as mitosis. Meiosis 1 was a little different because of the pairing of homologous chromosomes, which does not occur in mitosis. So during prophase 2 of meiosis 2, you just have a pair of sister chromatids, and then metaphase 2 in meiosis 2, the chromosomes align along the metaphase plate. They are attached to opposite poles of the spindle, and then during anaphase 2, now the sister chromatids will separate. They will move towards opposite poles of the spindle, and then telophase 2, again, the nucleus, it divides, so cytokinesis occurs, and you form 4 haploid cells. So the whole process of meiosis, you're going from 1 diploid cell to 4 haploid cells. All of these 4 haploid daughter cells are genetically unique from each other and the parent cell. And this is meiosis. So now I'm going to talk about the functions of mitosis and meiosis. So 3 main functions of mitosis is for asexual reproduction, is also for growth in multicellular organisms, and is also for cell replacement in multicellular organisms. The main function of meiosis is to reduce the number of chromosome sets by half, so that when gametes fuse during fertilization, a diploid zygote is formed. So meiosis only occurs in sexually reproducing organisms only, and it produces haploid cells. Your gametes in humans are haploid, such as eggs and sperm. These are haploid. And haploid cells, these gametes fuse together, so an egg and sperm fused in fertilization, which produce a diploid zygote. If meiosis did not reduce the chromosome number by a half, then your cells would die, so that would be just useless. So it's very important for the gametes to be haploid. So this is the main function of meiosis. So this is mitosis and meiosis.